All right, we're rolling. All right, I'm Porter Thames. I'm 22 years old. I'm majoring in biology and minoring in chemistry. And what's your name?
I'm Chandler, 24 years old. I'm studying community family addiction sciences.
All right. Anything else?
No,
Okay. All right. Well, today we are going to be talking a little bit about the sodium-potassium pump in each one of our cells, and a little bit about the experience really that I've had in my lab in cellular biology.
great.
So, all right. So the sodium potassium pump, I believe it's called the NTP, or no, sorry, not NTP. It's the NA/K ATPase. That's where that where the TP came from. But it's an enzyme that's found in all of our cells, and what it does is it takes a part of salt. It's dissolved in water. Sodium. It's the sodium ion. The other one's chlorine, and it takes it out of the insides of our cells and pumps it on the outside to create a varying gradient that and potassium comes into the cells while sodium's going out, and it creates a gradient that the cell can use to do other processes, and it's energetically favorable for the cell. Does that make sense? Kind of.
Absolutely.
Okay. So it's important. It's important because energy is not created, but I guess kind of created from the reaction itself, bringing the sodium in and the potassium out. It creates that electrical charge, and it's particularly important to muscle cells and to nerve cells. Kind of talked a little bit about that earlier, but each the pump brings out 3 sodium ions and brings in 2 potassium ions, and each one of those ions has a charge, and the charges are equal. So it's a positive 3 going out and a positive 2 coming in. So that difference in charge creates an electrical gradient, and it can causes contraction in muscle cells, or impulses in nerve cells, and with diseases, if you have an imbalance or a deficiency in potassium, which the pump needs to bring into the cell, it's going to affect those muscle and nerve cells first, probably. I do not know that, but it's in the plasma membrane of the cell, which is basically it's the wall, the fluid layer, really, not wall, that's surrounding all of our cells, and the specific focus of ours in the research project that we were doing was to check to see if varying mutations in the pump itself, which really that means like varying structures. There's little changes in the structure of the pump that causes it to do different things, and all throughout the animal kingdom, there's different mutations in the coding for those pumps, and it, like I said, it causes them to do certain things, and and it helps certain animals adapt to different uh, external situations.
That makes sense.
So different amounts of salinity and salt. But yeah, so we looked at the brine shrimp, and we were presented with a little information that the brine shrimp has a couple mutations that aren't found in other animals, and the brine to the pump, and um, the shrimp, they live in the saltiest, not the, like the saltiest place in the world, but I guess the saltiest place that animals live in. So they live in an environment that's like super salty. Uh, Other fish, if you put them in there, they would probably die instantly because they can't handle that amount of salt in the water. Um, and So I guess I should kind of backpedal a little bit. The pump is a protein, and proteins are made of amino acids, and proteins are basically just long sequences of amino acids that they get folded up, especially and by other proteins, and then once they're completely folded, they're able to carry out these super specific processes in the cell. And so most animals have, well, I guess all animals have a general specific sequence of amino acids that codes for their pumps. But some have different mutations, which are just different amino acids in place of the normal ones, which, like I said earlier, causes a different structure, and because of that different structure of the pump itself, it changes the function. Um, but the cause of the brine shrimp had that, they have that mutation, and um, their pump is able to well, it's thought that their pump is able to I don't know if I'm wording this right, but their pump is able to pump out more sodium, or sodium more efficiently. Hey, calm down. And she's just going crazy. Um, and and so it's a topic of our research to figure out what exactly is going on there. And so basically how we did that was we we the DNA for human pump has been sequenced in the Human Genome Project, and other pumps have been sequenced as well. There's salmon, shrimp, humans, all that's been identified and sequenced. And in our lab, we took various sequences of DNA, some mutated and some just regular DNA, or some with the mutations, um, and we were able to put that DNA into bacterial plasmids. We were able to replicate it with a technique called PCR, and so that basically means we've got a little bit of DNA, and we were able to use some enzymes and the building blocks of DNA to create more DNA. And then we took that DNA, and we injected it into bacteria, which there was some special treatments there with the bacteria, but that's kind of irrelevant. So then that bacteria, we allowed them to grow overnight or over a couple days, and make more bacteria, or I mean more DNA, and I guess bacteria, and
Mm.
we extracted the DNA from the E. coli to sequence to make sure that we got the mutations that we were looking for, and uh, that we did, and we didn't, but I guess we didn't do that either. Um, but Dylan and Pablo linearized the successfully mutated DNA and transcribed RNA from that, which is DNA turns into RNA, and then RNA turns into proteins, or that information is used to create proteins or the pump. Um, and so we took that RNA and put it into like a frog oocyte, which is basically just like a big cell. We use those because they're easy to observe and manipulate. And so the frog eggs took that information and created the pumps that we wanted to use. And uh, as the cells expressed more and more pumps, or more of those proteins, I guess we didn't do anything then, but after they were done, um, we used a technique called voltage clamping to measure the activity of all these different mutations that we induced. Um, and so it's pretty cool stuff. A bunch of that probably sounds like gibberish to you. That's okay. But oh, some of that made sense. But so that's pretty much what we did with the research, and all that happened in the cellular biology lab. But um, now I'm going to talk a little bit about the lab itself, and uh, in the lab exercises, I had a great time learning about the different procedures. And that I'd never done before, and practicing different procedures and techniques that I had done before. And it's always good to practice, and oh my gosh, we're worthless, to practice and to own your skills, I guess. And so I think a big part of that, maybe not a big part of that, I guess practice just in general is what makes you better. But something that helped a lot with that was I was in a lab with just a couple people. There's only 5 of us. Hey, calm down. Hey, calm down. And we had a couple instructors, and so it was, one instructor with 5 students, and so we got like a lot of one-on-one time, and that was really helpful, and I think it contributed greatly to everyone understanding what was going on, and and just learning and having a good time. But doing the actual research experiment, that was the first time I've ever done anything scientific like of that magnitude, and um, like taking DNA and mutating it, and then like expressing that DNA. I mean, I've read about it, but definitely never done it before. So that was really cool, and getting to meet Dr. Artigas, Pablo, and all of the people doing research with him. That was also really cool. I got to talk to them about about their experience, doing like graduate level research, and uh, they all seemed to love it, and I thought it was really cool. I thought a lot of it I did understand. There was definitely some things that I didn't. Uh, I didn't really get the voltage clamping, you know, very well. But that's also, I think I have like a working understanding of it, but I haven't taken physics like we've talked about. Uh, So that was kind of a setback there. But the concept of an electrical current, it's not too bad. I looked it up earlier, and I think I get it. Um, and the other thing that was kind of hard for me is understanding what the different mutations are, because and how not just what they are, but what that means and what they do. So yeah, so it was a good lab. I really enjoyed it, and it went by too fast. But I'm grateful for the experience. I look forward to hearing what everyone else has to say. So thank you, Chandler.